Today's topic is our gene expression. How we can study particular gene expression in a tissue that is explained in this video. So there is a one technique that is gene microarray that enables researcher to study all of the genes whether expressed or not in a tissue very fast. So in this technique, microarray or gene chip is utilized, which is created with the use of small glass microscope slide. It is just a small glass microscope slide. Now, single-stranded DNA molecule are spotted on this slide using the arrayer. It is a computer-controlled robotic arm, and this arrayer that will fix this particular single-stranded DNA, which fixes DNA at different spot on the slide. So, this multiple copy of complementary DNA that can be fixed over this small glass microscopic slide, and that is recorded by computer. So, that is simple way to study of this gene expression. Now, how this array is working, that is explained. First, we have to extract mRNA from a tissue of interest. So particular what are the tissue we are studying, and from that tissue mRNA is extracted. From that mRNA, complementary DNA is synthesized with the help of reverse transcriptase enzyme. So what are these complementary DNA synthesized from this mRNA? Again, it is labeled with a fluorescent dye. So what are the tissue we are learning? From that we will produce a complementary DNA which is labeled with a fluorescent dye. Now this labeled complementary DNA is incubated overnight with the array. Now this array, when it is incubated with the overnight with the array, it hybridizes with a different spot on the array. That array that contains complementary DNA sequence. Now that different spot which is containing different DNA sequence, and that is incubated overnight with this complementary DNA, and if there is a complementary sequence is present, then it will be hybridized. So these can have over 10,000 spot on this DNA array, which is once it is incubated overnight, then that sample will be washed out, and after washing that will be scanned by a laser that causes complementary DNA hybridized to array to the fluoresce. So if complementary DNA which is incubated overnight over this array, if it is hybridized, then it will fluoresce, and that can be detected with the help of particular computer. So fluorescent spots reveal which gene in this particular tissue were regulated, and intensity of fluorescence indicate relative amount of the gene expression in that particular tissue. So with the help of this microarray which is produced over that particular mRNA in the form of complementary DNA is added, and that complementary DNA that can be detected, and that is useful to indicate whether that gene is expressed, or intensity or amount of gene expression that can be utilized for the gene expression. In detail of this gene expression that is explained in this next picture. So first we have to isolate mRNA from the tissue. So then mRNA is converted into the complementary DNA molecules. So that is with the help of reverse transcriptase enzyme. Now that all is labeled with a fluorescent dye. Here that blue color dye is utilized. Now over that particular this complementary DNA which is produced from tissue mRNA, that all will be added over this microarray chip or the gene chip. Now that incubated for the overnight. So in overnight incubation, hybridization will occur. So this hybridization that is next that is detected. So there is a segment of chip which is containing more than 10,000 spots. Now that all overnight hybridization, if complementary sequence is present, then overnight incubation will lead to the hybridization of that particular complementary DNA of the tissue to the over that microarray chip. Now this what are the overnight incubation that rinse all excess of this complementary DNA and put the microarray in a scanner to measure fluorescence of each spot, so that each spot or this microarray will be measured. Then fluorescent intensity indicate the amount of gene expressed in the tissue sample. So over that corner that particular microarray chip will be looped. So in this image, blue spot indicate bright fluorescence and white spot indicate no fluorescence, and each will be read out. So bright fluorescent indicate highly expression of the gene in the tissue, while moderate fluorescent will indicate low gene expression, while light or there is a white color, no fluorescence, gene not expressed in the tissue sample. So simple, whatever the tissue which we are learning, and over that mRNA is extracted, and with the help of that mRNA we can detect whether that tissue or particular cell is expressing particular gene or not. So with the help of this microarray technique, it is very easy and convenient technique for the detection of that particular tissue is expressing that gene or not. So that is simple technique to understand about the gene expression, and it is all about the microarray technique.